Welcome to Peptides Today, the podcast dedicated to tracking the cutting edge of peptide science. I'm Sonia Rao, and if you're not keeping pace with the latest developments, you risk falling behind in critical research. Today, we're diving into a breakthrough peptide, C8, targeting Toxoplasma Gondii. This research could redefine our understanding of intracellular parasite control, offering novel strategies for future investigations. We'll explore its unique dual action mechanism and what it signifies for the broader field of anti-parasitic discovery. Stay tuned to ensure your research remains at the forefront.
Toxoplasmosis research is vital. It impacts host organisms globally and presents complex scientific challenges for study. Current research methods have limitations, requiring novel compounds to effectively control the parasite. Intracellular parasites like T. gondii modify host processes, complicating efforts to study their biology. The C8 peptide emerges as a compelling investigational agent against T. gondii in laboratory studies. This peptide offers a pathway to potentially superior control, enhancing anti-parasitic drug discovery research. C8 distinguishes itself through a sophisticated multi-pronged mechanism of action within host cells. It directly inhibits parasite invasion, a crucial step in preventing initial cellular uptake by T. gondii. This preemptive strike drastically reduces the parasitic burden observed in vitro models. T. gondii often manipulates host cellular processes, including critical defense mechanisms like autophagy. C8 restores host autophagy, a vital cellular defense mechanism against intracellular pathogens. By rearming the host cell, C8 enables a more robust intrinsic defense against the parasite. This dual intervention reduces the likelihood of resistance development compared to singular MOA agents. Preclinical studies show C8 exhibits potent parasitical activity across multiple T. gondii strains. Data indicates C8 significantly prolongs survival in acute infection models without host cell cytotoxicity. This favorable safety profile is a substantial advantage over existing investigative compounds in studies. Its efficacy across multiple strains suggests broad-spectrum activity, vital for globally prevalent pathogens. Early data for this peptide looks promising, balancing efficacy and safety during early research.
Fascinating insights into C8's foundational research, aren't they? If you're finding this deep dive valuable, make sure to follow me, Sonia Rao, for more detailed analyses. And for comprehensive updates across the entire peptide research landscape, you absolutely need to subscribe to Peptides Today. Coming up, we'll shift our focus to the rigorous development pathway ahead for C8, examining the extensive preclinical work and regulatory considerations required for its advancement. We'll explore the scientific hurdles and future research directions.
The journey from preclinical discovery to an approved research agent is arduous and long. For C8, the immediate next steps involve rigorous lead optimization and comprehensive preclinical development. This entails expanded efficacy, safety studies, and pharmacokinetic profiling in animal models. Exhaustive toxicology assessments, including acute and subchronic studies, are essential for compounds. Given its peptide nature, stability and degradation pathways are critical to characterize for research. Specialized formulations may be necessary to improve bioavailability and peptide half-life in studies. The article notes a need for chronic infection efficacy optimization, a significant research challenge. T. gondii forms dormant tissue cysts. C8's ability to target these forms is paramount. This requires specific in vivo models designed to mimic chronic infection and its reactivation. After successful preclinical work, an investigational new drug application supports human research trials. This submission consolidates all preclinical safety, efficacy, and manufacturing data for review. C8 might enter Phase 1 human trials within 3-5 years with robust funding. Advancing through later phases depends on demonstrating superior efficacy and a pristine safety profile. A strong regulatory strategy focusing on specific research populations could expedite its path. The C8 peptide represents a highly promising candidate in the fight against Toxoplasma Gondii. Its novel dual-action mechanism addresses critical limitations of current investigative therapies. The significant unmet research need provides strong impetus for continued investment and scientific study.
This journey through the C8 peptide's research potential highlights the dynamic nature of anti-parasitic drug discovery. It's a testament to innovative science addressing complex biological challenges. If you appreciate these deep dives, please follow me, Sonia Rao, for more detailed analyses and pipeline tracking. And of course, keep Peptides Today bookmarked for all your essential peptide research updates. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.